Dear students, today our topic is about the cell cycle. So, firstly, we discuss what are the cell and what are the key roles of the cell division. So, cell can be defined as the ability of organisms to reproduce, best distinguishes living things from non living matters. So, the continuity of life is based upon the reproduction of cells or cell division. Cell divisions is integral part of cell cycle. So this is the overview of the cell cycle which start from different phases and then end toward the mitosis phase. So these all steps we will discuss one by one with their functions and their role in the cell division. So the first step in the cell cycle is the interphase. So interphase include further three subdivision which is the G1 phase, S phase and G2 phase. So now we define what is the interphase in the cell division. So interphase is the period of the cell cycle that occurs before the mitosis. Basically mitosis is the phase of the nuclear division. It is the longest part of the cell cycle during which the cell is actively living, growing, and preparing for the division. Interphase is often mistakenly called the resting phase, but the cell is highly active metabolically. It means its purpose is to duplicate all the necessary cellular components so that two genetically identical daughter cells can be produced during the mitosis and subsequently cytokinesis. So that's why the interphase is divided into the three subphases. So first we discuss the subphase of the interphase which is the G1 phase. So here the picture can also showing that the recovery from the previous divisions occur in the G1 phase. Here the cell doubles all its organelles, cell grow in size and accumulate all the raw materials from the DNA synthesis which we call the DNA replication. So G1 phase is also known as the gap phase because the cell grow and function normally, protein synthesis is high and the cell increase in size. So the cell accumulates the resources needed for the DNA replication. If the cell does not receive a signals to divide, it may exit the cycle and enter a state which called the G0 phase. So the second phase of the interphase is known as the S phase. So S phase is the phase where the DNA replications have been occurred. And here all the proteins associated with the DNA are synthesized. So S is known as the synthesis phase where DNA replications, meaning the cell synthesize a complete identical copy of all its genetic material. So the amount of the DNA in the nucleus effectively double. Each chromosomes in duplicate and consist of the two identical sister chromatids joined at the centromere. In animal cell, the centrosome which organize the mitotic spindles are also duplicate. So the second phase which we call the G2 phase and also known as the gap 2 phase. Here between the DNA replication and onset of mitosis is the G2 phase. The picture also showing the G2 phase which is between the DNA replication and the onset of the mitosis. Here cells synthesize all the proteins necessary for the mitosis or the division. So the cell continued to grow and synthesize the proteins and all the organelles needed for the division, such as the components of the mitotic spindles. The cell check the duplicate chromosomes for any errors and make any necessary repairs. Upon successful completion of the G2 phase, the cell is fully prepared and committed to enter the M phase. The picture showing the M phase is the mitosis phase. So now what is the mitosis phase? So the mitosis phase include the two parts, which is the karyokinesis and the cytokinesis. Karyokinesis mean karyo meaning the nucleus and kinesis meaning the division. This is the first step of the mitotic phase and involves division of the cell nucleus. Basically, the primary goal of the karyokinesis is to ensure the accurate and equal distribution of the replicated genetic material, which is the chromosome, into two daughter nuclei. Cytokinesis. Cyto meaning the cytoplasm and kinesis meaning the division is the second and final step of the M phase, and it involves the division of the cell's cytoplasm. Basically, this process physically separates the two newly formed nuclei and the rest of the cellular content, such as the cytoplasm and organelles like the mitochondria and ribosomes, into two distinct daughter cells. So now this is the just overview of the mitosis, which is the somatic cell karyokinesis is with mitosis and occur in a series of the stages. So first we discuss what is the importance of the mitosis. So the significance of the mitosis is that it permits the growth and repair mechanism. In plant, it retains the ability of to divide throughout the life of the plant. In mammals, in mitosis necessary to fertilize the egg become an embryo. The embryo becomes a fetus and allow a cut to heal or a broken bone to mend. So it's the significance of the mitosis which play an important role in our cell division. So now we explain the mitosis phase, which is the prophase or the first phase of the mitosis. So in this phase, all the chromatins condenses into visible chromosomes. The nuclear envelope breakdown. So this we explain with the pictures here, that in the picture the chromatins is condensed. Here you can see in the picture the chromatins are the thread-like structure. Secondly, the centrosomes separated, moving to opposite ends of the nucleus. For example, the movement of the centrosomes occur and it move toward the opposite sides of the nucleus. So the centrosome starts to form a framework used to separate the two sister chromatids called the mitotic spindles. This is made up of the microtubules. Here show that the mitotic spindle is made up of the microtubules, so it helps to side by the centrosome toward the site of the nucleus. Fourth one is that the nucleolus disappear. In this case, it's mean it break down itself, and the nuclear envelope is disintegrates. It mean the nuclear envelope break, and so all the events occur in the prophase, which is the first step of the mitosis. So in the second step, which is before the metaphase, is the prometaphase. Simply in the definition, prometaphase or the metaphase is that where the chromosome align at the center of the cell, which is the metaphase plate. So this we can explain with the example or the picture here or the diagram showing that here the nuclear envelope fragmented. First, you can observe that the white color is the nuclear fragments here, which is have been fragmented. Secondly, all the chromosomes become more condensed. For example, the chromosomes appearance is more thicker as compared with the previous picture. So here you can see that all the chromosomes are condensed. Third, a nitochore is formed at the centromere. The middle point here you can observe that this is the nitochore. So a nitochore is formed the point where the sister chromatids are attached. You can observe that all the sister chromatids are attached with the nitochore. Now, all the microtubules attach at the nitochore. All the microtubules in the surrounding like thread-like structure attach with the nitochore, so they will start attachment and thickness of the chromosomes occur in the prometaphase. So it's mean all the chromosomes are ready for separation. The third step is the metaphase. The third step showing that all the chromosomes align at the center of the cell, which is the metaphase plate. So in the diagram here you can see that all the chromosomes align in axis called the metaphase plate. The axis showing is the metaphase plate, and note that all the spindles consist of microtubules. Here you can observe that all the microtubules attach to the chromosome and they start them to separate down. Okay, the first step is the anaphase. It's showing that all the sister chromatids separate in the anaphase and move to the opposite pole of the cell. So the diagram showing here that each centromere splits making two chromatids free. Here you can see that all the chromatids are free and move toward opposite poles. So each chromatids move toward a pole and cell began to elongate. It's mean cell increase their size caused by microtubules not associated with the netochore. Here the microtubules are not associated with the netochore, but it only work to elongate the size of the cell. Last step in the mitosis is the telophase. Telophase is basically a new nuclear envelope formed around the two sets of the chromosome at the poles. So here the picture showing that the formation of the nuclear membrane and the nucleolus occur. Here you can see that this one is the nucleolus forming the white coloring, or at the middle where the metaphase plate was, that is the cleavage furf occur, and the nuclear envelope forming. It's mean short and thick chromosomes began to elongate to form a long and thin chromatin. It's mean that the short chromosome become the long and the thick chromosome become thin chromatin, right? So in that case all the events have been occur, and the most important event, which is the formation of the cleavage furf, you can see the middle of the diagram. A shallow groove or a shallow nick have been appear in the cell near the old metaphase plate. And now the cytokinesis, which is the division of the cytoplasm, the two nucleus will become separate from each other. So this is the last step of the event of the mitosis, where the two cells become separate from each other. So this is the result of the mitosis. Here you can observe that the two daughter nuclei will be appear, each with same number of chromosome as the parent cell, just like the 2n in the parent, so the 2n also in the daughter cell. That's why they are genetically identical to each other and the parent cell. So these all are the steps of the cell division, which starting with the three different phases, and then we covered the mitosis with the four different stages. And now the significance and result of the mitosis have been shown that the each with the same chromosomes and genetically identical to each other and with the parent cells. So this is all about the cell division. Thank you so much.